Hey guys, this is Malinki. Welcome back to my channel Voice of Malinki. Today we will talk about gram staining, and if you are new in my channel, please subscribe my channel, and if you like my video, please do like, comment and share my video. So gram staining is basically a technique. It is a technique. It is a staining technique that differentiates between gram positive and gram negative bacteria. So we have all heard about gram positive bacteria, gram negative bacteria. Now, how they are distinguished or how they are named as gram positive and gram negative bacteria. Okay, so to understand that thing, first we need to understand the structure of gram positive bacteria and gram negative bacteria. So this is the gram positive bacteria. Now this bacteria has a very thick peptidoglycan layer in the cell wall, because we know that bacterial cell wall is made up of peptidoglycan layer. Now this gram positive contains very thick, this red part I have represented here as thick peptidoglycan cell wall, and this is the plasma membrane that is present beneath the cell wall. In case of gram negative, you can see that the peptidoglycan layer is very thin. So this red part is peptidoglycan layer, that is very thin layer, unlike gram positive bacteria. And one more thing, it has extra, a lipid layer. A lipid layer is present outside the cell wall. It is present outside the cell wall. So that is also extra in gram negative bacteria, because in gram positive bacteria you will not get any extra lipid layer outside the peptidoglycan cell wall, and the plasma membrane is present beneath the peptidoglycan cell layer. Okay, next what we can do in this staining process, the first thing, we put primary stain. We add primary stain, that is crystal violet. Crystal violet, so this stain, from the name itself you can understand, this crystal violet stain, it makes purple color to the bacteria, and this is primary stain. Primary means because it is the stain that helps us to distinguish between gram positive and gram negative bacteria. So it is the primary stain, main stain. So we have applied crystal violet stain, and you can see that crystal violet stain will color the peptidoglycan layer. So this cell is looked like purple, and this gram negative is also looking like purple. Both cells are stained by the crystal violet, and both cells are looking like purple. So if in this stage you look these cells under microscope, you will not get any difference. All cells are looking like purple. Next we are adding mordant here. So mordant is like a fixative. So in this case we are using Gram's iodine solution. This solution or this mordant, what it does, it actually fixes the color, fixes the crystal violet stain in the cell. Now after this stage also, both cells, both gram positive and gram negative, they look same. They both are purple color, and here also if you see these cells under microscope at this stage, you will see all cells are purple. Next we will add decolorization, that is alcohol. We can use 70% alcohol here. So we will use the decolorizing agent or alcohol. Now in this stage, what will happen? First we will talk about this gram negative. So in this stage, the lipid bilayer, this is the yellow part, lipid bilayer, that is present outside the cell, will be dissolved because of the alcohol. This lipid layer will be dissolved in the alcohol. So this lipid layer will be washed away. And when the lipid layer is washed away, the next layer, that is the peptidoglycan layer that is present here, this layer is exposed. And this peptidoglycan layer is very thin layer. And in this layer, the stain was fixed. Now, as this layer is very thin, the stain will also be washed away. As the stain is washed away, the cell looks colorless because all the stains are now washed away along with the lipid bilayer. The cell looks colorless. In case of gram-positive, it does not have any lipid outer layer. It has only very thick peptidoglycan layer, and in this layer the stain is fixed, and as this layer is very thick, the stain will not be washed away. So this cell looks purple or as it is under microscope. Now if you observe these cells under microscope, gram-positive cells look purple, but gram-negative cells are now colorless, so you can't see any gram-negative C under microscope because they are now transparent, colorless. How could you see them? So you need to see gram-negative bacteria also. That's why you will add one more stain, that is the counter stain. It is called counter stain, that's why. Now you can add safranin. Safranin gives red color to the cell. Now both gram-positive and gram-negative cells will take this safranin, but gram-positive cells are already look like purple. They are having very dark stain. Now although they are taking the red safranin color, but they will look like purple only. Gram-negative cells, which are colorless, which are transparent, will take up this safranin stain and will be looking like red. So if you observe these two cells under microscope now, the gram-positive cells are like purple and the gram-negative cells are red. That is the difference between gram-positive and gram-negative cells.